Yes, my dear students, did you got this point? Two channels, sodium channels, potassium channels. Look for the sodium channel in the diagram and look for the potassium channel in the diagram. And there is one pump that is called sodium potassium pump. The axolemma, outside it is called ECF, that is extracellular fluid. Inside it is called intracellular fluid. Outside the membrane is positive. Why? Because there is more of sodium ions. Inside it is more negative because it has more of potassium and electronegative protein. So this is a charge of a axolemma when it is resting. At the resting potential, this stage is called polarized state when outside is positive, inside is negative. Now, the resting potential is how much? Minus 70 millivolt is a resting potential of axolemma. Clear? Now, a stimulus is reached. That is a polarized state. If you look, polarized stage or the resting stage is impermeable to sodium but slightly permeable to potassium ions because to maintain the positivity of the membrane. Resting membrane potential is minus 70 millivolt. Who maintains this minus 70 millivolt? Sodium potassium pump maintains this minus 70 millivolt is an important question. Minus 70 millivolt is important question. Sodium potassium maintains it is an important question. Next, depolarized state. After minus 70, now the membrane is resting. If it neuron receive a stimulus of plus 10 millivolt, when it receive a stimulus of plus 10 millivolt, from minus 70 it will reach to minus 60 to minus 55 millivolt. Clear? At this point what happened? Sodium channels open. At this point what happened? Sodium channels open. When sodium channels open, look into the diagram, the sodiums will move out from outside ECF to ICF. When minus 55 to minus 60 millivolt is reached, sodium channels open. Sodium channels which are outside more will come into inside. Clear this point? Influx. That cause a depolarization of the membrane. What will happen at that moment? The positive will become negative. Whatever is a positive outside will become negative, and whatever the negative inside will become positive. There's a change, there's a depolarization. This stage is called depolarization, and then information is carried. Current is produced here. Next, what is repolarization? This sodium channels open. If you look into the diagram, at minus 60, what will happen? Sodium channels open, the depolarization takes place. The depolarization is such a way for how long this sodium channels open until the voltage reaches to plus 35 to plus 45 around. This sodium channels remains open. So much of sodium is moving inside, causing the positivity of the membrane. The point where it reaches to minus plus 35 to plus 45, sodium channels closes, but potassium channels open. Now, what is the aim? The sodium channel were coming from outside to inside. Now, since the positivity has reached to plus 35, it has to become negative now. So, more of potassium will move out. From where? From inner ICF to ECF, because potassium channels open at plus 45 to plus 35. It should be open until it comes to minus 70. But what will happen? This potassium channels opens for longer time, and it opens such a way for longer time that it leads to minus 90 millivolt. This stage is called hyperpolarization. It has to repolarize coming to minus 70. But since the channels open for a longer time, it comes to minus 90, that is hyperpolarized state. From minus 90, it has to come to minus 70, which is a resting potential. To come from minus 90 to minus 70, who is going to help? Sodium and potassium channel is going to pump, not channel, pump is going to help. By use of ATP molecule, from hyperpolarized state to a resting potential, a pump is used. ATP is used to bring that minus 90 to minus 70. But whereas sodium channel and the potassium channel, there is no use of energy. There is no use of ATP. That is what I have mentioned in the last class. Now I want you all to please read this one time. Everybody, I have explained you. Please read, and if you have doubts, do let me know. I'll explain you more. Come on.
Excuse me, ma'am.
Yes.
Ma'am, could you please explain that graph part again?
See, Sanika, minus 70. Can you see minus 70? That is a resting potential of a membrane. Okay, your membrane is resting. Resting means what? Positive outside, inside it is negative. If the stimulus that is received is plus 10 millivolt, stimulus receive one neuron पे. Okay, some messenger came and stimulated the neuron. When minus 70 को plus 10 से minus करेंगे तो कितना होगा? Minus 50 to minus 60. Okay, at this range what will happen? Sodium channels open, which are present on the exolemma. When sodium channels open, look into the diagram. When sodium channels open, the sodium will move inside from ECF to ICF. During that time, what happened? More of sodium is moving inside. Now that before it was a negative inside. Now since the sodium is moving, it becomes positive. Outside it become negative. So the changes of the charges, that stage is called depolarized state. And then this channels will open until what time? Until the potential reaches to minus plus 35 to plus 45. After it reaches there, the sodium channels closes and potassium channels open. After potassium channels open, what will happen? From outside, potassium channel is present where? Inside ECF. From ECF, it moves towards outside. From ICF to it moves towards the ECF, potassium ions, so that it can come from plus 35 to minus 70. But since potassium channels open for the longer time, more and more sodium goes off and it reaches to minus 90 millivolt. But our aim is to get to minus 70, which is a resting potential. The purple color, that is minus 90 stage, is called hyperpolarized state. So sodium and potassium pump, if you look into the diagram, it will influx. It will influx 2 potassium, outflux 3 sodium to maintain the positive outside and negative inside with the help of one ATP molecule. Important. They'll ask you how many sodium outflux, how many potassium influx, how many ATPs are used, who maintains the minus 70 millivolt sodium potassium pump, who brings the hyperpolarized to polarized state sodium potassium pump. Getting the point? Now read. I want you all to read what is given. You will understand. Please read.
Ma'am,
Yeah.
can you explain me the exolemma point?
Exolemma is a neurolemma. What is exolemma? Axon. You remember? Neuron. See, this is a cyton, right? This is the axon, right? This is a dendrites. Do you remember the structure we studied?
Yes, ma'am.
Yeah, this membrane is called axolemma. On this neuron, this is one neuron, okay? From one neuron to other neuron, the information need to pass, right? How it is passing? It is passing through polarized state, depolarized state, repolarized state. Polarized state, depolarized state, repolarized state. This is how the information from one neuron to other neuron is passing. Getting the point? That's the reason generation and conduction of impulse. We say that axon, that neurons transfer the impulses, the information. How does they do? By the help of sodium potassium channels and sodium potassium pump. Getting the point? Yash,
Yes.
is it clear? Sania, Sanika, is it clear? Padma, is it clear? Please read, please read. See, it's easy. Read, and still if you have doubts, I can explain you. You have written these notes, please read. You already have written the notes, read. Can you see this? Positive outside, negative inside. I have drawn only this diagram there. हमने सिर्फ इतना ही draw किया है. This part. See, we have just drawn this part. Has a diagram. We have just drawn one part, right? Negative inside, outside positive. So if you look here in the depolarization, kya hota hai? Positive will become negative, negative will become positive. Again, repolarization, what should happen? Negative should be outside, inside, and positive should be. But here, what will happen? More of hyperpolarization takes place. Between the depolarization and repolarization, there is a hyperpolarization state where more of negativity, it goes to minus 90 millivolt. We want to get back to minus 70 millivolt. To maintain from minus 90 to minus 70, who plays a role? Pump plays a role. By removing 3 sodium and 2 potassium by use of 1 ATP molecule, it comes from minus 90 millivolt to minus 70 millivolt, which is a resting potential. Then again it will receive the stimulus. Again from minus 70, it will become minus 55 to minus 60 millivolt. Sodium channels open. Then it will reach to plus 35 to plus 40. Then potassium channels open. Then potassium channels open. Then again, polarize to repolarize, depolarize, depolarize to repolarize. Got the point? Any difficulties? Very easy. Come on, quick. Have you all gone through it? Yes, during depolarization stage, the potassium channels open and potassium will move from inside to outside. Not during depolarization. Yes? Yes, it is not during depolarization. During repolarization, see, depolarization graph clearly says that at minus 55 to 40, what will open? Sodium channels open. So only sodium will move. How you can say potassium is moving? Only sodium is moving through channel. There is no use of energy. It's a passive movement. After it reaches to plus 35 to plus 40, then sodium channel close, potassium channels open. Then there is repolarization, which is bringing by potassium is moving from inside to outside. Yes, got the point. The graph clearly says everything. Very easy. If you look, you understand the graph and this diagram. Very easy to write this diagram. Write that diagram which I have given you, and also this graph. Explain it for 5 marks. You will get your full marks. Others, any doubts? Padma, any doubts? Prabhu Desai, any doubts? Can we move further? Same thing. This is okay. Look to this diagram, everybody. Shifa, see. Resting potential, who maintains? Pump maintains. Depolarized state, voltage channel, sodium channel open. Repolarized, potassium channel. Resting potential, again sodium and potassium channel, pump. If you want, you can take a click. Very easy. No difficulties. Again, same. Okay. Next. After this, we are moving to. So if I ask you what is the resting potential, tell me what should be the resting potential of the resting membrane of neurolemma. Minus 70 millivolt. Very good. At what voltage the sodium channels closes? At what voltage the sodium channel closes? At what voltage the sodium channel closes? Very good. At what voltage hyperpolarization happens? At what voltage the hyperpolarization happens? Very nice. Now please write down this transmission of impulses. Please write down this. Done. Please write down and then I'll explain you.
And what is the second part of synapse?
Sorry? Transmission of impulse, synapse, presynaptic membrane of presynaptic neuron, synaptic cleft, postsynaptic membrane of postsynaptic neuron. Done. Okay, next. Write down this. Done? Can I explain you? Can I explain?
Yes, ma'am.
Okay, now listen. Here,
Say
now
for
we have said, wait for a minute. Okay, do it quick.
yes, ma'am.
Okay, now we know that the impulse are conducting. For example, this is one neuron. Okay, this is one neuron. So we have studied now how the impulses are traveling to the axolemma, right? And here this is another neuron. Now it has to transfer this message which is coming through the axolemma need to be transferred to the next neuron. How it can happen? It can happen by two waves. But before understanding this, we need to know what is synapse. Very important question. Synapse is a junction between two neurons. It's a junction from this is one neuron, this is second neuron. So this junction is called synapse. Understood? Hello. Did you understood?
Yes, ma'am.
Next. Synapse has include how many parts? Three parts. One is presynaptic. This is neuron 1. This is neuron 2. Right? Presynaptic membrane. This membrane is called presynaptic. For example, in this case, this is the presynaptic membrane. This is postsynaptic membrane. And the gap in between is called synaptic cleft. So presynaptic membrane, postsynaptic membrane, and the cleft together it forms one. This is A, B and C. Together it forms synapses. Point is clear to all? Yes. A nerve impulse is transferred from one neuron to another neuron through the junction called synapses. Yes. Now next. There are two types of synapses. One is chemical synapse, other is electrical synapse. What is chemical synapse and what is electrical synapse? At electrical synapse, electric current flows directly from one neuron into the other. Means if in electrical synapse, what happened? If there is one neuron here, the other neuron here, it directly, continuously is transfer the message from here to here. There is no intermediate here. There is no somebody to carry the message from one neuron to other neuron. It's a direct transfer. That is called electrical synapse. Electrical current flow directly from one neuron to other neuron. Impulse transmission across two electrical neuron synapses is always faster because there is no carrier. It goes directly from one neuron to the other neuron to the third to fourth, like that. But what happened in chemical synapses? Let me tell you. In chemical synapses, if this is one neuron and if this is one neuron, this is a synaptic cleft. In between, there is something called neurotransmitters. So the information from here is transferred to the neurotransmitter. From neurotransmitter, it is going through the third neuron. So, which is a faster, electrical one or chemical? Tell me, which is a faster? Hello, are you there? Electrical one. Because there is no in between. There's a faster than the chemical synapses. Why chemical synapses? Because in chemical synapses, there's a neurotransmitters which takes part. Clear? Okay, fine. So I hope you understood. Okay, read about this. Quickly read. Come on. Read what I have explained you just now. Quick, just a few seconds. Done? No doubts, right? All clear. If you have doubts, you can ask me. I'm assuming there are no doubts. Now, draw this. Draw this. Are you all there? Done?
Wait, ma'am. Ma'am, please wait.
And do let me know once you are done. Okay. Done, everybody?
Done, ma'am.
Okay. Now listen to me. Now the impulses have reached to the end. When the impulses have reached to the end, I have told you the presynaptic cleft. Look into the diagram when I'm saying the terms. When I say presynaptic cleft, see to the presynaptic membrane. When I say cleft, look into the diagram for the cleft. So there is a presynaptic membrane, there is a cleft, and there is a postsynaptic membrane. This all three will form synapses. This I have told you. Now, the impulses that are coming from the neuron 1, when it reaches to the end, at the knob of the neuron, there is a vesicles. Those are called synaptic vesicles. These synaptic vesicles contain neurotransmitters. How many neurotransmitters? 2000 to 10,000 neurotransmitter in one vesicle. This number is for one. Are you getting? One vesicle will have 2000 to 10,000 neurotransmitters. So there you have many many synaptic vesicles. Now, when the impulses reach to the end, the calcium channel is open. This calcium channels goes and fuses with the synaptic cleft, bring this synaptic cleft and allow it to fuse with the presynaptic membrane. Then it will release the neurotransmitters into the synaptic cleft. On the postsynaptic membrane, they are the receptors for these neurotransmitters. They bind to the receptors. As it binds to the receptors, in the second neuron, the sodium channels open and the information is carried further. Got the point? Now, please write down this. You will understand it better. Start writing this.
Ma'am, what is written after voltage?
Voltage gated calcium channels open.
Okay, ma'am.
Done? Okay, please read through it. Any difficulties there? These neurotransmitters can be excitatory neurotransmitters or can be inhibitory neurotransmitters. Means it can allow the next neuron to get excited based on the information it is coming from the brain, or else it can allow the impulses to stop there. It depends, okay, whether to send the impulse further or not. It can be excitatory or it can be inhibitory.
Ma'am, this is of chemical synapse, right?
Yeah, we are not talking about electrical synapse. That's the reason chemical synapse includes synapses, right? A neurotransmitter. So it should be a chemical or electrical when there's a neurotransmitter.
Chemical, ma'am.
Yes. Done, everyone. Have you read through it? Any difficulties there? See, action potential reaches the axon terminal. Calcium channels open. Calcium causes the vesicles to release neurotransmitters. Neurotransmitters crosses the synapse. Neurotransmitter binds neuroreceptors and triggers a signal in the postsynaptic membrane. Very easy.
Ma'am, is the screen frozen?
Screen is frozen. Okay. Can you see it now? Okay. You do not have to draw this. We have explained this part. Next. When you talk about the
Excuse me,
neurotransmitters.
ma'am.
Yeah.
Ma'am, can you go back to the previous note slides?
Yeah. When I ask you why, don't respond. When I ask you, are you done? Are you done? Done?
Done, ma'am. Thank you.
Next. Now, these neurotransmitters can be excitatory neurotransmitter or can be inhibitory neurotransmitters. Among the excitatory neurotransmitters, acetylcholine, epinephrine, glutamate, dopamine. All these are excitatory neurotransmitter. Please write down.
Ma'am, could you please tell again?
Sorry? Screen is frozen. Just a minute.
My screen is not visible.
Wait a minute. Now it's visible. Write down. Done? Now, the decrease in this any of the neurotransmitter can lead to some of the diseases. Like if it decrease in dopamine, it leads to a disease called Parkinson's disease. Decrease in dopamine or increase in dopamine leads to schizophrenia disease. All are brain related diseases. Okay. Decrease in acetylcholine lead to Alzheimer's. Done? Next one. Inhibitory neurotransmitter. You should know which are the excitatory neurotransmitter and which are the inhibitory neurotransmitter. GABA, which is gamma amino butyric acid, glycine, serotonin, dopamine. Dopamine is act as both excitatory and inhibitory neurotransmitters. Huntington's chorea is due to the decrease or the deposition of gamma, that is gamma amino GABA, that is gamma amino butyric acid.
The screen is frozen, ma'am.
Okay, fine.
No, ma'am. It was visible.
Just a minute.
Yes, ma'am. It was visible.
It was visible. Okay, take it.
Excuse me, ma'am.
Yeah.
Ma'am, Huntington's chorea is due to deposition of hormone.
Due to deposition of GABA in the neurons.
Okay.
Done. Can we move to the next? Let's move to the next. Okay. Now, next we'll be studying about this is all about the neuron transmission. Now, next we'll be studying about human nervous system. Write down human nervous system, which is categorized into central nervous system, peripheral nervous system. Central nervous system is brain and spinal cord, whereas peripheral consists of motor neurons and sensory neurons. Under motor, you have somatic autonomic nervous system, somatic nervous system, and autonomic nervous system. Under autonomic, you have sympathetic and parasympathetic. All this we are going to study in detail, right? Is it screen frozen? Can you see the
Yes.
nervous system?
Yes.
Yes,
No,
ma'am.
ma'am.
It's frozen.
Okay, 2 minutes. Check it now. Please write down. Sania's screen is black. Is it black
Yeah.
to all?
No, ma'am, now
Like
it's
this.
okay.
Okay.
Ma'am, screen is not visible.
Can you see the screen?
Yes.
Okay, please write down.
No, ma'am.
Ma'am, the slide is not visible.
Slide is not visible. Now is it visible? Can you visible now? What is this one? Sanjana can see it. Why not others? Okay, please write it down quickly.
Ma'am, now I can see the team screen.
It's visible to others. So can you please check, re-log in?
No, ma'am. We can see the team's screen.
Yes, ma'am, the same is me.
Yes, ma'am.
Can you see? Sanjana has shared that slide. Can you please copy from there? Okay, nervous system. Can you all write down which is Sanjana has shared it on the chat box about the nervous system? Central nervous system, peripheral nervous system, brain, spinal cord, neurons. Okay, I'll be back soon. Start writing, which is on the chat box. I'll be back 2 minutes. Okay. Done writing that part? Is it done writing? Can we move further? Now, first, central nervous system consists of what? Brain and spinal cord, right? Central nervous system, let us write down. Okay. Central nervous system consists of brain and spinal. Let us discuss about central, then we'll go to peripheral, okay? This both has some common character, four common character. So we'll study first four common character, then we'll study about brain and spinal cord. Okay, we'll be studying about the 4 common characters. So which are the 4 common characters that is found in brain and also in the spinal cord? Let us study that. The 4 common characters are, first one is presence of 3 meninges. First one, presence of 3 meninges. Single, it is called meninx. What is meninges? Which is nothing but connective sheath of connective sheath. Right? Very important to note, fishes have single meninges or meninx. Amphibians and reptiles and birds, 2 meninges, the connective tissue. Whereas mammals, very important, you should know this, many times of questions, including human, 3. So 1, 2, and 3. Clear? As I said, there are 3 meninges. Let us draw the diagram. Yeah, after the diagram, I'll scroll up. Let me draw this first. Done? Please do it. Anika, done? Very good. So human brain has how many meninges? Tell me how many meninges are present in the human brain? One, two, and three. So it is pia matter, arachnoid matter, and dura matter. Clear? What is the function of meninges? The function of meninges is protection. Protection. So the matter here is pia matter. Then come arachnoid. Then comes dura. Here you need to remember from inner to outside. They'll ask you the order. Inner to outer. Easy way to remember is pad. PAD pad. And pad is the three meninges which are present. This is a trick. Clear? And CSF is, what is the full form of CSF? CSF. Cerebrospinal fluid. Done? All clear?
Ma'am, then where is the blood-brain barrier present?
Blood-brain barrier we'll discuss later. It is present in the brain. We have studied the neurons, right? Neuroglial cells. In the neuroglial cells, those are which cells of the neuroglial cells forms the blood-brain barrier? Hello. Which cells of the neuroglial cells forms the blood-brain barrier? We studied in
Astrocytes.
Very good. Astrocytes. Remember? Yeah. You write down this explanation for this will be given in the next class. Okay? Are you done writing? Any doubts for today's class? Did you understood well? Scroll up. Did you all understood well? Is anybody in the class who have not understood today's lecture? Understood? No more queries? Everyone? That's really nice. So, can we wind up for today's class? See you all in the next section. Until then, what you guys has to do? What do you need to do?
Keep revising.
Keep learning. Bye-bye, everyone. Take care. Yes, keep revising.
Keep revising. Thank you, ma'am.
Keep learning. Okay, bye-bye.